Hello, everybody. I know it's cold and icy outside, but I wanted to start with this picture of a UT Austin student catching the OChem wave. I made this by AI yesterday, and I thought it was just kind of funny. Okay, what we're going to do is we're going to talk about vision. And I started talking about this last week, but as you're going to see, there's kind of a surprise ending here. Just to recap, what I told you was that this molecule here, cisretinal, attaches to a protein called opsin via a Schiff base. And the key to that is a reversible linkage. Notice this cis double bond. When a photon hits this molecule, it turns out that it creates a situation where the antibonding orbital inside that bond is now populated. Essentially, you're breaking the double bond, the pi bond there. That allows the molecule to rotate and form the more stable trans geometry. That trans geometry means the whole molecule has a very different shape. Instead of this bench shape, it's now a linear shape. That movement triggers the opsin molecule to then send a signal to the brain that says a photon was absorbed. This is essentially how we see. What's interesting then is because this Schiff base is reversible, after the photon is absorbed and the molecule, the retinal molecule, turns into the all trans form, that molecule dissociates. It is then recycled to make more of the cis form version, which then bonds once again via the Schiff base to a new opsin, waiting for the next photon to hit. You've probably all experienced this when you get a flash of light or it's too bright out. You can't see for a moment. And that's because all of your cis bonds turn to trans and you're waiting for the molecules to recycle. In other words, dissociate because of the reversible nature of that imine bond formation and then turn back into the cis where it can then rebind to opsin. Okay, but there's something about this that is really pretty cool. And it has to do with where does this molecule come from? So let's talk about that. What I need to do is I need to give you a bit of a flashback to something we talked about last semester, the terpenes. Now, everything I'm about to say is not going to be on an exam, so don't worry about that. But I do think it's really interesting, so let's talk about it. Terpenes are molecules that are made in plants, and that's the thing to remember. Plants make really interesting molecules, some of which you're familiar with. For example, this limonene is what it gives lemon and orange its flavoring. There's some molecules you're familiar with in perfumes, such as lilac and especially lavender, or menthol, a flavoring and that you've probably been familiar with in a lot of different products. It comes from peppermint. These molecules all look pretty similar, but they're even more similar than you think, because plants make their complex molecules starting with what we refer to as isoprene. Isoprene is a 5-carbon unit that has the one branch, the one methyl branch. And actually, it's not the isoprene with the double bonds that Mother Nature uses in plants to build up the molecules. It's these two here. You're going to learn all about this in biochemistry. The point I wanted to make, though, is that even though these molecules down below here, they look related, they're more related than you think because they're all based on putting two of these isoprene units together. There's biochemical machinery that is involved in this in all the different plants. But we can look at this by highlighting those 5-carbon units. And when we do that, you'll see that all those 4 molecules down below can be made by assembling these 5-carbon units, these so-called isoprene units. Molecules that are found in plants like this are referred to as terpenes. And why am I saying this? I'm saying this because if we go back to look at the molecules of vision, what you'll notice is that cisretinal is actually a terpene. It's composed of 4 of those isoprene units. This is an incredible observation. Why? Well, turns out terpenes are from plants. So how is it that it's the part of the key sight or vision mechanism within mammals? Well, it's because we co-evolved with plants. It turns out our bodies cannot make cis retinal. Instead, we eat it and it comes from plants. Pretty cool. So let me tell you exactly where it comes from. It comes from these two main sources. You can get it in meat that you eat in the form of vitamin A, or you can get it directly from plants. This is beta carotene. Beta carotene, as you might expect from the name, that's what gives carrots their orange color. So you may have heard your mother say at one point, hey, eat your carrots, it helps your vision. She's telling you the truth. Because it turns out our source for cisretinal is actually vitamin A that comes from a variety of sources, most notably plants, or beta carotene, where this bond is cleaved biochemically inside your body to transform it into vitamin A, which then, if you notice, this alcohol turns into the aldehyde for all transretinal. Then inside your body, it's then converted to the cis-retinal, where it can then be localized within the opsin protein. It'll diffuse in. So that's essentially the process. You start by eating carrots and or ingesting vitamin A, then that's converted to the aldehyde in your body, and eventually the cis double bond is made. Just to show you that those other molecules are terpenes, I've highlighted the 5-carbon isoprene units in beta carotene, vitamin A, and then the trans, and then the cis retinal. So I hope you found that interesting. I think it is incredibly cool that our vision, mammalian vision, depends on molecules that we get from plants. And if you know how to look for the signature, namely the 5-carbon isoprene unit, you can immediately recognize that cisretinal came from plants. So I'm going to leave you with an image that I made by AI that'll help you remember this story. I asked the AI generator to give me a cute bunny with a huge eye so you can remember how vision, mammalian vision, is related to carrots, because of course our bunny is eating the carrots. I hope you found this useful.